So this is the second part of cholera. In the first part, we have talked about the classification of the vibrios and the pathogenesis of the cholera infection, and the clinical manifestations or the clinical features of cholera. Now we will talk about the laboratory diagnosis of cholera. So in the laboratory diagnosis of cholera, we have to first see what is the specimen that we collect for the diagnosis, because in any diagnosis we have to first collect the preferred specimen. So the preferred specimen that we collect in case of cholera infection is the freshly collected watery stool. Okay, in a universal container, you will see the universal container in your practical microbiological examinations. So the preferred specimen is the freshly collected watery stool. Other than that, we can use the rectal swab in case of asymptomatic cases. We remember that in 75% cases there is no symptoms. That means asymptomatic cases occur in about 75% of persons who has got infection with the Vibrio cholerae. So in such cases we have to collect the rectal swab. Other than that, we can also collect the vomiters. Okay, now after collection of the specimen, we have to transport the specimen to the laboratory. So for transporting the specimen till the laboratory, we need some transport media so that the bacteria can remain viable inside the during the transport. So for that we can use any one of these four media, and one should must remember these all four names, because any one of these name may be asked in MCQs, or these transport media for Vibrio cholerae are very commonly asked in the Viva examinations as well. So for the transport of a specimen, the different transport media that we use are the Venkatraman Ramakrishnan medium, the alkaline peptone water, and the Carey Blair medium, and the autoclaved sea water. These are the four media that we use for the transport of specimen in cholera suspect. In what ratio do we transport the specimen? So for that, we transport about 3 ml of water stool into 20 ml of the media. So suppose if we are using this Venkatraman Ramakrishnan medium for transport of cholera fecal specimen, then we will take 20 ml of this Venkatraman Ramakrishnan medium and 3 ml of the water stool. We will put this water stool into that Ramakrishnan medium, and then we'll transport the specimen. Next comes the direct microscopy. So after the collection of the specimen, first we try to detect it directly under microscope after gram staining. The first step that we perform is the gram staining of the specimen. So with the specimen, we prepare a smear on a clean grease free glass slide, and then we gram stain the slide. After gram staining, we can see whether there is gram positive bacteria or gram negative bacteria. So if it is Vibrio cholerae, then we will be able to see the gram negative comma shaped bacilli. Okay, and it has got a characteristic appearance. This appearance is very important to remember, which is called as the fish in stream appearance. Fish in stream appearance. It will look like as if the fishes are swimming in the river. Something like that will be the appearance. So that is called as the fish in stream appearance. After that, we will perform the motility testing, and for motility testing we use the hanging drop method. That is most commonly used method from long ago. So we will perform this hanging drop method. How do we do that hanging drop method? So for the hanging drop method, we will first take a cover slip. Okay, this is the cover slip, and we will take a drop of specimen over that cover slip, and then at the margins of the cover slip we will add some vaselines. Okay, at the margins of the cover slip, then we will take a slide which has a concavity on it. Okay, something like this. A slide will take, and then we will put this slide over the specimen in this way, so that the concavity just lies over the drop of specimen. Okay, and the this Vaseline gets attached to the uh, concave slide. Okay, after this, okay, let me remove all this. Okay, so after that we will invert this whole system, and after inverting it will look like this. Okay, see, the cover slip will become the cover slip will come at the upper side, and the concave slide will become below the cover slip. Okay, and we will see this under the microscope, and we will see, when we see it under the microscope, we see motile bacteria in that drop of specimen. Okay, so that means the bacteria is motile. We get a clue that the bacteria is motile. Now in case of Vibrio cholerae, we see darting motility. This should also be remembered, that darting motility is seen in case of Vibrio cholerae. Darting motility means sudden rapid movement. Okay, this sudden rapid movement is called as darting motility. What is meaning of sudden rapid? uh, at one point of time you will see that the bacteria is here, suppose here, and suddenly you will see that it has moved to here rapidly. Okay, so this is called as the rapid movement. You will very clearly see if you prepare this hanging drop method very cautiously, and under the guidance of professors. Next we do is the culture. So for culture, first we have to do the enrichment broth. Okay, so first we do the enrichment broth. For enrichment broth, the stool sample is inoculated on the enrichment broth to increase the number of Vibrio cholerae and inhibit the commensals. What is the use of this enrichment broth? So the use of the enrichment broth is to increase the number of the Vibrio cholerae and to inhibit the commensals, because we know the gut of the human has many commensals bacteria. So we want that they do not interfere into the results. So for that we have to inhibit those commensals which are coming into the fecal sample along with Vibrio cholerae. Therefore we use this in enrichment broth, which inhibit the commensals and increase the number of Vibrio cholerae. So what is the enrichment broth that we use? Okay, these are the two enrichment broth, that is alkaline peptone water and the Monsource taurocolate telluride peptone water. These are the two enrichment broths that we use for the for the for growing the number of the Vibrio cholerae, that is for enriching the um, specimen. We use these two. These two names are also again very important. One should must remember these names. Next is the culture on the selective media. So next is culture on the selective media. So different selective media we use. One of that and the very important of all is the TCBS agar. TCBS means thiosulfate, citrate, bile salt, and sucrose agar. The Vibrio cholerae produces here yellow colored colonies. These colony characteristics, these colony characteristics as well as the media must be remembered always. Okay, these yellow colored colonies on TCBS agar, this should must be remembered. Next comes the alkaline bile salt agar. Okay, next is the alkaline bile salt agar, on which the Vibrio cholerae produces oil drop colonies. And other than that, we have the Monsource gelatin taurocolate telluride trypticase agar, MGTTA. MGTTA. So on this Monsource gelatin taurocolate telluride trypticase agar, the Vibrio cholerae produces translucent colonies with grayish black center. After that we have to prepare a specimen, we have to prepare a smear on a clean grease free glass slide, and then we will gram stain the smear. So smear is produced with the colonies that has been produced on any of the selective media, and then we will gram stain it. After gram staining we will be able to see the characteristic feature of the Vibrio cholerae, that is gram negative comma shaped bacilli, that is the gram negative comma shaped bacilli. Again we will perform the motility testing by hanging top method, and we will be seeing the darting motility. Next is how will we identify this Vibrio cholerae. So for identification we have some conventional methods, and we have also got some automated methods like Meltitov and Vitek. So in the conventional test we perform these five tests. Okay, we perform these five tests, that is the catalase test, oxidase test, citrate test, urease, and the indole test. How will we remember these tests? So we remember these tests by the Cocui score. COCUI, put it here. COCUI, C for catalase, O for oxidase, C for citrate, U for urease, and I for indole test. P3NP is the formula for Vibrio cholerae. That means first P3 means 3 positive, upper 3 are positive. See, 1 2 3, upper 3 are positive. Then NP, the fourth one is negative, the fourth one is negative, and then again P, so fifth one is positive. So by this Kokui score, uh, we can remember the conventional results of the conventional tests. I have this is my way to remember the conventional test of different bacteria. For Vibrio cholerae it is P3NP. For Vibrio cholerae it is P3NP. And for look, if you are looking for this result, for this conventional test results of other bacteria, then you can see my video on the UTI, okay, urinary tract infection, where I have talked about the uh, about the conventional test results of different bacteria in this same Fukui score manner. That will be very easy for you to remember all the conventional test results for different bacteria if you remember by this Kokui score. So Kokui score for Vibrio cholerae is P3NP. Now coming to the string test, that is also a method of identification. So what is done in this string test is a colony of Vibrio cholerae is mixed with a drop of 0.5% sodium deoxycholate, and then as we mix it with sodium deoxycholate, the suspension become mucoid. Okay, and once the suspension becomes mucoid, when it is lifted with a string, then it forms a, sorry, when it is lifted with a, it will not be a string, it will be a when it is lifted with a probe or with a ring. Okay, so please change this, it will be a ring. Okay, so when it is lifted with a ring or with a inoculating loop wire, then there is formation of string, and when the string is found, that is called as the string test positive. This string is nothing but a thread like thing is formed. Suppose okay, wait, suppose if this is a glass slide, and this is where you have mixed the colony of Vibrio cholerae with 0.5% deoxycholate, and you have got this inoculating loop with you. So when you touch this touch this suspension, when you touch this suspension with the inoculating loop, you will see that a string is formed like this. Okay, this is called as the string. Okay, this is called as positive string test. So this is in with in case of the Vibrio cholerae. Next is zero grouping. So for the zero grouping, we have got some O1 antisera. So if there is agglutination with O1 antisera, that means it is O1 zero group. If there is no agglutination with O1 antisera, then test with O139 antisera. And if now there is agglutination present, then we can say it is a Vibrio cholerae O139 zero group. If again it is if again agglutination is absent with O139 antisera also, then it is a non O1 non O139 zero group. Okay, so by that, using the antisera, we can differentiate between the serogroups. Next we have the biotyping. How do we do the biotyping? So biotyping is done in case of O1 serogroups. Why it is important? It is important because the classical biotype has caused 6 pandemics out of total 7 pandemics, while the recent pandemic of cholera, that was the seventh pandemic, that was caused by the E1 tor biotype. That's why it becomes very important to differentiate between them. So to differentiate between the classical and E1 tor biotypes, as we have talked in our earlier video, 4 types of test that we perform is VP test, then susceptibility to the polymyxin B, and then susceptibility to the group 4 bacteriophage, and then also we can also see the beta hemolysis on C blood agar. These 4 characteristics differentiate between the classical and the indoor biotypes of the O1 serogroup of Vibrio cholerae. Other than that, we can use the molecular methods to uh, diagnose the Vibrio cholerae, because molecular methods are far much advanced techniques and very easy to perform. So PCR detects the gene of the Vibrio cholerae toxin, and other than that, we can also use the biofire film array. So the gastrointestinal panel can detect the pathogen, and then last but not the least, we have to perform the antimicrobial susceptibility testing for detecting the drug resistance of the Vibrio cholerae, so that we can provide with the best antibiotic for the treatment. Now, the prevention and control. Prevention and control again is a very important topic about the cholera, because it is a hygiene related disorder. Okay, so how can we prevent this cholera? So for prevention, we have got some general measures, some chemoprophylaxis, and some vaccines as well. So in general measures, we have some administrative measures and some individual or the personal measures. In the administrative measures, if the administration provides safe water supply, if the administration improves sanitation in a locality, if the administration investigate any outbreak very promptly, and if the administration notifies the disease if there is any case occurring into the block or into the locality, then the cholera can be controlled. So these are all the administrative measures, because these are done by the administrations. What a individual can do to prevent the cholera? So an individual can maintain the food hygiene and perform frequent hand washing to prevent cholera. Okay, then we come to the chemoprophylaxis. So chemoprophylaxis is given along with the chemoprophylaxis is given by the tetracycline, but this chemoprophylaxis is given only to the members of family, that also during the epidemic. So if the epidemics occurs, then this tetracycline chemoprophylaxis is given to the members of family. Remember, the chemoprophylaxis for cholera is never given to the travelers going to cholera endemic area. This is not like malaria, where we give chemoprophylaxis for any person who is going to the malaria endemic area. Okay, so it is not like the malaria. In case of cholera, never ever chemoprophylaxis is given for the travelers who are going to the cholera endemic areas. Okay, this is in contrast with the case of malaria. Next comes the vaccines. So vaccines were initially of 2 types: the injectable killed vaccines and the oral vaccines. Now this injectable killed vaccine has been removed from the market. It is no longer used, and we are left with only the oral vaccines. We are left with only the oral vaccine. In the oral vaccines, we have got some killed whole cell vaccine and the live attenuated vaccines. So in case of the killed whole cell vaccines, 2 oral doses are given, and this is indicated for individuals more than 1 year of age, while the live attenuated vaccine is a single oral dose vaccine, and it is recommended for adults of 18 to 64 years of age. That is the difference between the killed whole cell vaccine and the live attenuated vaccine. So this is how we can prevent or control the cholera. So in prevention and control, we have got the general measures, chemoprophylaxis, and some vaccines. And this is all about the diagnosis also, uh, lab diagnosis of the cholera. If you write this much in the exam, uh, in the exam, if the diagnosis of the cholera is asked, then it will be enough to fetch you almost full marks. And please remember these names. Please remember, please remember these transport media, then this appearance, then this motility of the bacteria, then the enrichment broths, these enrichment broth names. Please remember these names, whatever I am marking with blue. These names are very important in case of cholera, or as far as the microbiology is recommended. The cholera chapter itself is very important, and in the cholera chapter, these points which I am marking with blue pen are very important. These selective medias, and this color of the colonies, these selective medias and the color of the colonies, these selective medias and the color of the colony, these are very important to remember. Please remember this, whatever I am marking with blue. And these conventional test results are also very important. You can remember simply by Kokui score of Vibrio cholerae, that is P3 and P. Okay, so this is all about the lab diagnosis of the cholera. Next we will see the some short notes in the topic of cholera, which are important from exam point of view, and then the cholera topic will get to an end. That's all for today.